Hi there. Let's do a quick little review of excitation-contraction coupling in skeletal muscle. So what we're looking at right here is a pathway coming from the nervous system that would stimulate skeletal muscle fibers to contract. So in green here, what we have labeled is the cell body of a somatic motor neuron. A somatic motor neuron is the type of neuron that will send efferent messages from the central nervous system, most likely the spinal cord as illustrated here, to distinct targets outside in the peripheral nervous system. In this case, because we are talking about a somatic motor neuron, those targets are always going to be skeletal muscle fibers, as opposed to autonomic motor neurons, in which case their targets will typically be cardiac muscle, smooth muscle, glandular tissue, endocrine tissue, basically anything that is not skeletal muscle. So what you see here is the cell body of this somatic motor neuron within the gray matter of the ventral horn of the spinal cord. It projects its axon out through the ventral root of that particular spinal nerve, as all efferent messages do. And you see the axon of this somatic motor neuron extend out. But here's the interesting thing about somatic motor neurons. A single somatic motor neuron is not necessarily going to have just one single cell as its target. Further down towards the more distal part of the axon, towards the axon terminus, you can actually see that the axon splits off and diverges into, as I have illustrated here, 3 different directions, although it could possibly be many, many more. So this one single axon of this one single somatic motor neuron actually has 3 axon terminals. So what that means is that if a single action potential is fired through this somatic motor neuron, that action potential is going to split off into each of the 3 divergences from the original axon, meaning that neurotransmitter is going to be released from all 3 of these axon termini. And as we have seen many times before, this neurotransmitter, because this is a somatic motor neuron, is always going to be acetylcholine. And that acetylcholine is going to bind to nicotinic receptors located on the plasma membrane, or sarcolemma, of these skeletal muscle fibers. So we have these nicotinic receptors here that are all ready and waiting for that released acetylcholine to bind to them. So what you see here, the somatic motor neuron, including all of the muscle fibers that are under its control, comprise what is called a motor unit. What that means is that anytime we get an action potential going through this one particular somatic motor neuron, all of the muscle fibers that are within the control of that motor unit are going to contract simultaneously. Whereas other muscle fibers, like these 2 you see over here, that are not in the same motor unit, they will not contract unless they are contacted by the somatic motor neuron that is in charge of them. So all of the millions and billions of skeletal muscle fibers that are contained within a particular piece of muscle, each muscle fiber is going to be under the control of a certain motor unit. So if you, say, lift something that is not very heavy, you probably don't need to have action potentials fired through very many somatic motor neurons. You just need to have enough motor units activated in order to give you the contractile strength to lift whatever it is you're lifting. But if you are lifting something that becomes progressively heavier and heavier, you may need to recruit additional motor units in order to give you the extra oomph in order to lift something that is much, much heavier. So the more somatic motor neurons that you fire, the more motor units you get involved, the more skeletal muscle fibers contract, and a more forceful contraction will be had. So now let's review what actually happens from the time acetylcholine is released from these axon termini to the point where the muscle fiber becomes contracted. So let's focus in right here. So if this is one axon termini of that somatic motor neuron, and this you see right here is one single muscle fiber, then the first thing we need to start with is the release of acetylcholine in this region that you see right here, this invagination of the skeletal muscle fiber's plasma membrane, which is called the the motor end plate. So motor end plate. So like I said, this is just a kind of special invagination of the sarcolemma that is meant to accommodate that axon terminal, and it is highly concentrated, as we said, in nicotinic receptors that bind to acetylcholine. So nicotinic receptors, as we have discussed before, are always excitatory. They are always excitatory because essentially they are ligand-gated sodium channels. So when this acetylcholine, which is in the synaptic cleft here, which the cleft we actually will now call the neuromuscular junction, the special name for the synapse that we have between somatic motor neuron and muscle fiber, this acetylcholine is going to bind to our nicotinic receptors here, and as such, we are going to open sodium channels. So, as was the case with neurons, as we've seen many times before, when you open sodium channels, sodium will flow from high concentration to low concentration from the extracellular fluid out here to the intracellular fluid here, and the end result is we are going to have a lot of extra positive charges inside the sarcoplasm here, inside the intracellular fluid. That will produce depolarization of the muscle fiber. So the muscle fiber is just like any other cell. It has a resting membrane potential, which is negative, just like a neuron is. So if we make the inside more and more positive, we get depolarization. If we get enough depolarization, just as the case was with a neuron, we will hit a certain threshold and we will activate an action potential. However, an action potential in a neuron was conducted down the length of a very long axon. We do not have an axon with a muscle fiber, but what we do have is right here, this very long invagination, kind of like a big deep canal that penetrates deep into the muscle fiber. This is called a transverse tubule, or T-tubule. If we get sufficient depolarization, our action potential is going to be carried down the length of this T-tubule. So what does that get us, really? Well, notice that the T-tubule runs very close to the sarcoplasmic reticulum. So the sarcoplasmic reticulum serves almost exactly the same purpose that the endoplasmic reticulum does in any other type of cell, but of particular importance here is the fact that the sarcoplasmic reticulum contains a very large concentration of calcium. So a lot of calcium here, hardly any calcium in the intracellular fluid, meaning the sarcoplasm. So out here, very little calcium to be had. The problem with having so little calcium in the intracellular fluid out here is that muscle contraction, as we understand it, as a shortening of the length in between Z discs within the sarcomere structures, this cannot happen if calcium is not present. So really what muscle contraction ends up being about is somehow making calcium available for these fibers that are contained on the thin filaments called tropomyosin. Tropomyosin is a very long fibrous protein that basically winds itself along the thin filaments, the actin filaments, and blocks any sites that could be interacting with the thick myosin filaments here that you see in green. So as long as that tropomyosin is in the way, no interaction between actin and myosin can occur, and no muscle contraction is going to be possible. That is where the sarcoplasmic reticulum comes in. When we get an action potential, which originated from the depolarization we get from the acetylcholine, that action potential is going to open voltage-gated calcium channels on the SR membrane, meaning calcium is going to come flooding out. And when calcium comes out, that calcium will bind to another protein located near the thin filaments called troponin. And troponin, when bound to calcium, will actively move that tropomyosin out of the way, freeing up the myosin binding spots on the actin, and now suddenly myosin and actin can interact, and we can get muscle contraction in which the sarcomeres shorten and the Z discs come together. Now, muscle relaxation, once you have contracted the muscle, is just going to be the process of reversing everything that we have shown here. So the muscle relaxation will involve such things as getting calcium back into the sarcoplasmic reticulum, ATP exchange with the myosin heads, which, if you do a little research on your own, you'll see how that pertains to the concept of rigor mortis. And then repolarization of the muscle fibers to get any excess potassium that has formed due to the process of the action potential. You want to get the muscle fiber back to the resting membrane potential, because as we saw with neurons, if there is too much potassium in the extracellular fluid, that hyperpolarizes the neuron, hyperpolarizes the muscle fiber, and it's going to make action potentials and sustained contractions that much more difficult to do. Now, muscle relaxation and muscle fatigue are complicated, complex concepts. It's probably oversimplistic to just narrow it down to these three, but overall, this should be a pretty good review of some of the major topics and concepts involved in excitation-contraction coupling of skeletal muscle. So we'll go ahead and end it here for today, and I will see you next time.